Very good evening, everyone. Thank you for having patience and bearing with us. Once again, very good evening. I'm Dr. Himani, and on the behalf of Vital Biotech, Agriculture Forum for Technical Education of Farming Society, Kota, Faculty of Veterinary Sciences and Animal Husbandry, Ranchi, and Birsa Agriculture University, Ranchi, I cordially welcome you all to the third day of this training. This international training come workshop program is on a very innovative topic. The title of this training is Cutting Edge Innovations in Veterinary Science for a Sustainable Future. So before starting the training, I would like to let you all know a little bit about the organizers. So the organizers are like Vital Biotech. It is India's leading publication research and training institute. Its goal is to tackle the world's most pressing problems with which are related to agriculture, food, and the Agriculture Forum for Technical Education of Farming Society, Kota. It serves as a spiracle It serves as a spiracle between the network of rural communities in India and other stakeholders such as government, development organization, and corporate entities. Whereas Birsa Agriculture University, it was established on June 26, 1981 after its inauguration by then Prime Minister Srimati Indira Gandhi ji, and its objective is to develop area-specific technologies and manpower in the field of agriculture, animal husbandry, and forestry for the agricultural development of Jharkhand and economic upliftment of tribal and other backward classes of the region. The program and activities of teaching, research, and extension are being carried out in this university through 7 faculties and 12 colleges. Besides this, the university is also having one College of Biotechnology, one Center for Agribusiness Management, 6 directorate, 3 zonal research station and farms, and university farm and 16 Krishi Vigyan Kendra and and many more. So the university is well equipped with the state-of-art infrastructure and other service units. And whereas the College of Veterinary Science and Animal Husbandry, it was established on November 30, 1961, and initially it was a part of Ranchi University. Later on, it became a constituent of Rajendra Agriculture University in 1971, focusing on UG program. And in 1972, the PG programs are also added with expansion in 1977, and in 1981, it is incorporated in Birsa Agriculture University itself. So since then, this university is offering UG, PG, and PhD program and diploma and certificate courses also. This college is also involved in research research and education. So once again, I congratulate the organizing for organizing this insightful training program for all the veterinary professionals and for the field veterinarians as well. So today we are highly privileged. We have such honorable dignitaries among us. So dear audience, it is a matter of great pride that the first guest of today's session is Dr. Deepak Upadhyay. Dr. Deepak Upadhyay sir has joined, and before sir start his talk, I'll let you know a little bit about sir. So Dr. Upadhyay is an associate professor and head of the Livestock Production Management Department at the College of Veterinary and Animal Sciences. And this college is under Rani Lakshmi Bai Central Agriculture University, Jhansi. Sir holds a master's and PhD in veterinary sciences, and sir is recognized with various prestigious awards, including Academic Excellence Award, NEO MAC Project Sixa Award, and DST Inspire Fellowship, ICR SRF Fellowship, and Silver Medal for the PhD. The contribution of Dr. Upadhyay to breed conservation have led to the registration of Rohilkhandi, Bundelkhandi, and Chaugarkha goat breeds with ICR Breed Registration Committee. Dr. Upadhyay specializes in breed conservation, feeding management, dairy housing management, livestock health management, foraging behavior, phytochemical nano-formulation, and integrated farming system. With 29 research articles published in Stafford Journal, 3 books, and 126 other publications, sir has made significant contributions in his field. Sir is a life member of various professional society, including Indian Society of Animal Production and Management, Indian Academy of Veterinary Nutrition and Animal Welfare, and Indian Poultry Science Association. Sir has been involved in numerous prestigious research project and serving as principal investigator and co-principal investigators in these projects. And in these projects are related to goat improvement, livestock productivity, forage crop genetics, grassland restoration using remote sensing, drone technology, etc. Additionally, Sir is playing a vital role in teaching, administration, and extension activity. And Sir is also teaching UG and PG students and various subjects he is teaching and guiding them in their career. So, Dr. Significantly to the field of livestock production and management uh, has a vital role in this field. So today, sir is going to talk on a topic which is visible on your screen. The topic is strategies for sustainable management of livestock and poultry. Dear sir, now you can just unmute yourself and you can start your talk.
Deepak sir, you already unmuted. Your voice is.
अनम्यूट तो कर लो अपने आप में.
I think there is some technical issue with sir and sir is trying to.
अब हुआ है.
Hello.
Yes, sir. Audible, sir.
There are some issues, I think.
Yes,
Sorry
sir.
for
Yes,
that.
sir. You already unmuted that time.
No, actually, I logged in from my laptop now, but there was some problem in the laptop.
Okay,
Now
okay.
I am logged in from my mobile only.
Okay, okay, okay. No problem, sir.
Yes.
Some
So thank you so much. Shall we have this in full screen, please? Dr. Dinesh.
Dr. Dinesh, please make this presentation to slideshow mode.
Please unmute, Dr. Dinesh.
Well, it is shared in slideshow showing mode only.
Yeah,
No,
but
no,
it's
it's
It is in the, I think,
not a slide share mode. You can share it again.
reshare it.
Yeah.
Now it is visible.
No. It is not visible, doctor. It is visible, but it is in the not in that presentation mode actually. Slideshow mode.
Dinesh sir, please send me. I will do. I think sometime like that problem is there.
Yes.
Yeah, the technology we can never rely on.
We can rely, but we should not fully rely on that either.
Yes.
Okay. Thank you, ma'am, for the nice introduction.
Thank
And
you, sir. Now the podium is yours.
thank you so much. And I think
Now it's
88 participants have joined this meeting. And I congratulate the organizers of this 7 days international training come workshop on this cutting edge innovations in livestock, veterinary science for sustainable future. And as ma'am told,
sir,
Hello.
voice is.
Dr. Deep, yeah,
Hello.
you accidentally muted yourself.
Okay, but
Yes.
presentation अभी नहीं दिख रहा है मैम इसमें ना.
Yeah, slides slides are not visible right now.
Dinesh sir, slides.
Is it visible?
Not in slideshow mode.
I'm sharing slide. So
Yeah, you can share the PPT with Dr. Jitendra.
I send you on mail. Is it not visible?
Dr. Deepak, now you can unmute yourself. Your cost now.
Dr. Dinesh,
Thank
the
you, thank
PPT is
Thank you so
visible,
much.
but yeah, presentation is visible, but not in the slideshow mode. That's the problem. There is some technical issue with your system.
And Dr. Dinesh, you share your screen, not this presentation actually.
I
Share your complete screen, then you select, you open that PowerPoint in slideshow mode in your screen. I think you are selecting PPT, na?
No, I'm sharing a screen, then selecting PPT.
Okay. So maybe some problem is there.
Actually,
Yes, sir.
huh?
Try
So
to
till
send
this,
me. Try to
okay,
send
no
me.
problem, no
I
problem, sir.
already sent you
Till
mail.
you share this presentation, I start my presentation only. I will start with the introduction.
Yes, now it's fine. It's in slideshow mode.
Yes, yes. Okay, okay. So now we'll start. So the topic of today's talk is strategies for sustainable management of livestock and poultry. As we know that livestock and poultry production is in India is very, very important, and for the world also it is very, very important. But please, please next slide. But livestock and this poultry sector, they both the sectors are always blamed to have contribution in the climate change. And the according to FAO, we see in this slide that the global livestock emission amount to 7.1 gigaton of CO2 equivalent per year, which represent 14.5% of all anthropogenic greenhouse gas emissions. So this is a very big blame on the livestock sector. This that livestock sector is contributing to a very big problem of the globe, that contributing to the greenhouse gas emission. Further, livestock production is also blamed to have a major driver of the deforestation, especially in tropical region, to acquire the land for the fodder production and for the grassland development. Further, the livestock farming requires vast amounts of land for grazing and growing animal feeds. This places pressure on the ecosystem, leading to habitat destruction and biodiversity loss. Further, the water scarcity and pollution resulting from the livestock operations are critical environmental concerns. So we have seen that the livestock sector is contributing somewhere to the environmental destruction and environmental harm. It is associated with the environmental harm presently. So what further one more problem in present livestock system is, next please. Next slide please. Yes. So in India, if we see that India's milk yield as compared to the milk yield in the US. But if we compare the average milk yield of both the countries, then we will come to know that how inefficient we are in the production. This is the case scenario of the dairy production. But similarly, in case of sheep production, in case of the goat production, pig production, poultry production, or any other kind of livestock production, we are lagging behind as compared to the developed nation or the world average. So we have seen that these two things are existing in the scenario, that livestock is somewhere having some negative impact on the environment, and also our livestock production system presently it is not as efficient as the world's livestock production. So what we need to do, we have to consider here. So what is the next step? But is it so that livestock is only contributing to the negative aspect? There is no positive aspect? No, this is not right. Livestock, although have a bigger positive aspects than its negative aspect. Next slide, please. Next slide. No, previous one, yes. Livestock is essential to achieve the sustainable development goals. Sustainable development goals have been decided by the community of the world to develop sustainably. So out of these 17 sustainable development goals, livestock sector have the say in out of this 17, 8 sustainable development. Next slide, please. 8 sustainable development goals livestock is contributing. And what are those 8 sustainable development goals? Next, please. Yes. So here are that 8 sustainable goals to which livestock is contributing directly, like poverty reduction, hunger reduction, good health, gender equality, decent uh, work and economic growth, responsible consumption, climate action, life on land. So, livestock is contributing to develop the globe sustainably also, apart from having some negative aspect. Next slide, please. And also, livestock build the global and national economies. If we see the livestock sector, it is contributing on an average 40% of the agriculture GDP globally. And that percentage is growing continuously. Demand for milk and meat in Asia will be more than double by 2050 from 2010 level. So here we are seeing that the demand of the livestock food product is on increase also. So demand is there and it is increasing gradually. So livestock value chain provide also a large number of jobs. So it is also associated with the employment. And agriculture and livestock are the two pillars. If we talk about the Indian economy and especially the Indian rural economy, so agriculture and livestock, we all know that these are the two pillars which support our Indian rural economy. So we see in terms of employment, in terms of livelihood, in terms of the food product, and in terms of the agricultural GDP contribution to the total GDP. In all these areas, livestock is contributing to a major contributing a major chunk. We can see in here in this figure that out of the livestock employs 1.3 billion people involved, means that's 1 out of every 6 people is involved in the livestock sector. The livestock sector produces $1.27 trillion every year. And if the livestock sector was a country, it would be the 15th largest in the world in terms of the economy. So this say the impact of the livestock sector on our daily life and our economies, on our employment. Next, please. It is the new economy of the poor. And if we see in terms of our country, so if we see that the contribution of the livestock sector in the GDP is continuously increasing since 1980, and the contribution of the agriculture is decreasing. It again emphasizes the impact of the livestock sector. Although we see that not that much focus have been given to the livestock sector in previous years. Apart from that, livestock sector is growing tremendously and contributing tremendously. So we have seen that there are negative impact of the livestock. There are the negative, there is the lack of the inefficiency in the production in the livestock. Apart from that, livestock is contributing, livestock is giving very so many things to us and the globe. So we need to consider this aspect that livestock is important, food demand is increasing, the livestock product demand is increasing. So what we have to do, we have to harmonize the production. We have to tune this thing, that we have to reduce the negative impact, or we have to create some system where we we can have a lesser impact of these negative aspect, and more and more positive aspect can be created. So here comes the sustainable strategies. So what we have to do, we have to work sustainably. Next slide, please. Next, please. Yes. So here we can see sustainable livestock production. That is our topic. So what is sustainable livestock production? Sustainable livestock production is the linchpin that harmonizes the need for animal-based product with ecological well-being. So here we consider the environment first. We have the environment. So there are, you see in this next figure, here are 4 pillars, which are most important pillar of sustainability of the livestock farming. One is the environment out of this, another is economy, institutional, and social. So these 4 pillars are very, very important when we talk about the sustainable livestock production. So what we came to know, we have to harmonize the need for animal-based product with the ecological well-being. So it places a strong emphasis on sustainable practices, animal welfare, and financial viability. So all these things we have to consider here. In I think it is visible to all that this figure, there are so many things. We have people, we have planet, and we have the livestock or poultry. So with respect to each and everything, we have to see. If we talk about the people, we talk about the employment, we talk about the community support, we talk about the antimicrobial stewardship, food safety and quality, food defense, and we talk about the, if we talk about the animal, we talk about the livestock. So we talk about the productivity, efficiency, health, welfare, biosecurity, and for the planet, we talk about the use of the land, use of the water, the energy, the greenhouse gases, the waste, and all. So all these things we have to consider simultaneously, and we have to see the, we have to make the balance. Next, please. Yes. So here comes, what are the benefits of this sustainable livestock farming? Sustainable, we have seen the definition that we are making the balance. So it help to reduce the negative, previous slide, please. Yes. Help to reduce the negative environmental impact and conserve the environment. It aims to reduce the emission from the livestock sector, or we say poultry sector. It emphasized to improve the animal welfare by the ethical treatment. It optimizes the uses of resource, say water, feed, and reduce the waste and inefficiencies. So therefore, it can lead to a safer and higher quality livestock products. It enhances the resilience of farming community by diversifying the income, and it can satisfy growing consumer demand for sustainably produced products. So here we have seen that there are so many benefits of the sustainable livestock farm. So as our topic of today's is, what are the strategies with which we can convert this existing production system to a sustainable livestock farming system. So next slide, please. Here are the strategies for the sustainable livestock production system. So I have made a very, very important some strategies which can be covered in this topic. There are so many strategies by which we can convert the existing system to the sustainable livestock system. Out of that strategies, we will discuss few strategies here in this presentation. So first and foremost, what we have discussed in the very first slide, that the livestock is blamed to cause the climate change through the greenhouse gas emission. So if we have to sustain with this livestock system, and if we have to grow, which we need to grow because we need to increase our production to meet the increasing demand. So for that, we have to reduce the methane emission. Then further the water footprint. Now the water is becoming very important topic of the talk. So we need to see the what is the water footprint of our livestock food products as compared to other product, and we have to improve the water use efficiency. Third is we have to use the integrated approaches, say for the pest management. Likewise, we can use several integrated approaches to make our the existing livestock system sustainable. Further, the improving welfare. So we need to consider the welfare of the livestock. What is the negative impact of the intensive system? We have not considered the welfare of the livestock. But here, to make it sustainable, we have to have the care of the animal. We need to improve the animal welfare. Further, with the what technologies we can use, what are the existing technology and how they can be incorporated in the livestock production system so that it can be used for the betterment of the livestock production, for the increase in the productivity, for the increase in the welfare, and for the increase of the resource use efficiency. Further, the adaptation of the climate resilient technologies, say for the breeds and other things, we have to use the as per the climate resilience, considering the climate resilience in the mind. Next, please. So, first is to reduce the methane emission. So, if we need to decrease the temperature by 1.5 degrees centigrade, which is the target given by the IPCC, from ruminant must be reduced by 11 to 30%. So methane emission from ruminant should be reduced by 11 to 30% by 2030 and 24 to 47% by 2050 as compared to 2010 levels. So here is the target. We have to reduce the temperature. We have to stop the global warming. So we have to reduce the methane emission. So which can be what are our target? 11 to 30% by 2030. So what are the strategies available for reducing these, which can be classified? There are several strategies. So when I was making this presentation, I considered one review paper where I have seen that more than 99 technologies available, tested and published in the peer-reviewed journal about the reduction in the methane emission. And out of that strategies, they can be classified into three categories. One is animal and feed management. Second is diet formulation. And third is rumen manipulation. So for example, this 3-NOP, this is the one such supplement which has shown to decrease the methane emission in dairy cow by up to 30%. Some vaccination have been also tried. Some tanniferous forages have also been tried, and some electron sink like nitrate can also be tried. So next, please. Yes, here in this figure we can see our three categories, which are this is the paper Arndt et al. 2022. You can review. You can see that paper. Animal and feed management. In the animal and feed management, what are the things available to reduce the methane emission? Are the feed processing. You can process the feed to reduce the methane. You can select the genetically selection of the crops of the feed for the low methane production, improving the animal health, improving the pasture management. So if you are using the extensive system or the grazing system or ranch system, their improving pasture management have impact on the methane emission. Increasing the feeding level, if you increase the level of feeding, the methane emission comes down. Increasing the quality of forage, optimizing temperature, and total mixed ration feeding. These are some options available for animal and feed management. These are some promising options. Further, in the diet formulation, we can have the byproducts, we can decrease the forest to concentrate ratio, which will decrease the methane emission. We can provide the mineral and salt, more oil and fat, oil seed, increasing the protein, tanniferous forages which have high tannin content, and feeding urea also. Further, in third category is the rumen manipulation, where we can provide the additives. We can have a vaccination like defaunation. We can do. We can feed the material which can be used as a electronic sink. Next, please. So here it has been classified. These categories further can be classified into product-based reduction or absolute reduction. Product-based reduction means the strategies will decrease the methane emission intensity. Also, they will increase the production of the milk or growth, whatever it is. And absolute reduction means the strategies which will reduce the methane emission, absolute methane emission, but they will not affect the production. They will not decrease the production and they will not increase the production. So here we can also classify it like this based on the product based reduction or absolute reduction. So if we see that increasing the feeding level, so in next column we have seen the potential emission reduction. Here it is written the CH4IM, means CH4 production intensity for milk. It is reduced by minus 17%. And relevant production system, we can see this strategy is useful for both feedlot and mixed system and the grassland system also. Likewise, all the strategies have been classified here. You can see that decreasing the grass maturity means cutting the grass at early maturity. It will increase the quality of the fodder. So it will reduce the methane emission intensity for milk also, and by the minus 13%. And third one is the decreasing dietary forest to concentrate ratio, where we can see the methane emission intensity for milk and methane emission intensity for gain both have been that minus 9% reduction can be noted. Likewise, several strategies have been enlisted here. Here we can see the methane inhibitors, some tanniferous forages, electron sinks, oil and fat, oil and seed. And likewise, we have seen the potential emission reduction have been provided here. So you can see there are lot of options are available by which we can make the existing emission reduced to the reduced level by 2030, and target can be achieved. Next slide, please. Yes. So our next strategy out of apart from the this reduction in the methane emission should be to improve the water use efficiency. Now we are talking about the how much water we are using per unit of the livestock product. Say if you are producing 1 kg milk, so how much water have been used for that milk production? So we know that for what water is required for the livestock production for the drinking purpose as well as for the other purposes. Other purposes will include the washing and the for the system whatever we are using the water. So for drinking water, the water use is required in a very less amount, and that is 2%, and that is very essential that we have to use only. So most of the water is used to produce the feed for the livestock. So feed production, we have to see how we can reduce the water use. So it is necessary to introduce the alternative solution for the water supply in the facilities for dairy cows using separate supply system for watering and washing. So whatever the system we are using, we can change that system so that for the watering we have a different water line, and for the washing we can use the recycled water and other water, so that the water use efficiency will increase. For efficient process for milk cooling, wet washing and yard housing can add up to significant saving in the water. we can use the different kind of system for cleaning of the livestock sheds, which include the hydraulic dragging system for the waste. So if we use this hydraulic dragging system, it uses the hydraulic system which will push the waste apart from the sheds, and that will be using very less water. We can use the constant level drinker. Here in the figure, we have seen that these drinkers are now available, where the constant level is maintained with the use of this valve technology. So the water is maintained at a constant level, and we don't have to waste more and more water. The thing here is to use the water use efficiency to make the sustainable livestock production. Next, please. So one strategy I found here is the integrated pest management. In livestock farming, we are using several kind of drugs. So how we can reduce the use of these drugs? We have to see. So one kind of approach may be the integrated management of the pest. In farming, such the ectoparasite infestation like flies, ticks, and worms can cause a significant problem to the animal health. So which will result in the more and more use of the medicines. So we have to reduce by biological control, like dung beetles can help break down the livestock manure, which will ultimately reduce the breeding ground for the flies. So in this way, we don't have to use the medicines. Cultural practice like rotating, if you are using in a grazing system, we can rotate the pasture to disrupt the life cycle of the parasites, reducing their impact on livestock. Mechanical control, we can provide the fly traps which can be used around barns, while mechanical brushes are available, which can be installed in the livestock shed, which will help to remove the ticks from the livestock. Next, please. Improving the animal welfare. 1 minute, please. 1 minute. Hello.
Yes, sir, you are
Yes.
audible.
Yes. So for next strategy to make our livestock production sustainable is to improve the animal welfare. So what we are doing in our intensive system in poultry, we have seen that animals are kept in very restricted spaces. We are keeping them in the cages. For the intensive dairy production also, for intensive beef production also, we have seen such a pathetic condition of the animal, where animal are kept in very restricted space to save the space and to produce more and more product on it. But this is causing impact on the animal health, which ultimately reducing the production and also the quality of the product, because ensuring good animal welfare result in the healthier and happier animal, and ultimately this well maintained animal under the very good welfare condition will be producing very good kind of quality product for the consumer. So nowadays the awareness regarding the use of the product which are being produced by a farm where animal are being maintained in a very good welfare condition is increasing. So therefore, to as the income of the people will rise, this kind of demand will rise. So by improving animal welfare, we will have a very good quality product. So for this, we have to develop a kind of a system. Here I have indicated some system. First, in part of A, we have to first assess the existing production system for the animal health and welfare. So we can assess this based on the subjectivity of perception of stakeholders like farmers and veterinarian expertise, and based on the objective indicators like on-farm monitoring, precision livestock farming, sensors, etc. Then we will study about the positive and negative interaction, synergies and trade-offs of this livestock production, these welfare issues. Then, by using the existing technology, we can develop a new system with a good welfare. For example, now, if we are talking about the conventional housing system in the dairy, where animals are kept in the conventional barns with the concrete floor, so their animal is having a problem. There are more chances of lameness of the dairy animal, which will ultimately result into the mastitis and other problem, which will further reduce the life of that animal and reduce the production also, and also the quality of the milk, where somatic cell count will be more. So by accessing that system, changing the strategy, imparting some animal welfare implication, we can make a system which will be the more performing and more sustainable. Next, please. Next slide, please. Yes. So now we are living in IT age. We are interacting here in this training program in the online mode. So we are interacting in a Zoom. We are in our own space. Somebody will be in home, somebody will be in office. So using technology, optimal use of technology can also be done in the livestock sector, and that is called as precision livestock farming system, where various sensors are used, data analytics is used, and automation is used to monitor manage livestock more effectively. For example, it includes the wearable sensors. This device monitor vital signs like to measure the heart rate, the temperature, the activity monitoring, the stress sign, etc., etc. Automatic feeding system, yeah, robotic feeding system are also available. Monitoring with the remote sensors, posture monitoring, all these technologies, and now we know the thing is coming, the Internet of Things, IoT. So that IoT can also be incorporated and also being incorporated already in the livestock sector in developed country, although we are lagging behind, but we have also started to use these technologies like the RFID system, some monitoring system. Likewise, we are slowly coming and using this technology in the livestock sector, but need is we have to use more and more system. Next, please. Here in this image, we can see that to measure the impact, the ruminal gas production, the production of the greenhouse gases by the animal, by using the technology, we can incorporate in it in a figure A. You can see it is the feed bunk where animal. It is an automatic kind of feeding system where animal can go and directly with its tag, it will read the system. System will read and accordingly it will dispense the feed where animal can feed, and simultaneously we can measure the amount of the CO2 or methane emitted by the animal in this chamber also. And also there in figure B, we can see the capsule. The gas measurement capsule is available, which is having some sensors kind of thing, which can be placed in the animal's stomach and it will measure the uh, greenhouse gas production also. And tracer technology, it is a very old technology, SF6 technology to measure the methane emission, which can also be used uh, with the use of the technology. Also, we can use this technology to measure the gas. So, what I need to convey here that we have to incorporate the technology in the livestock production system so that it will be moving towards the sustainability. Next, please. Next, please. Yes. So I told you that we have to use the genetic resilient breeds, genetically advanced breeds. So genetically resilient breed means we can incorporate the local breeds. We can improve them. We can improve their efficiency and we can use them because they are more disease resistant. They are more climatic resilient. So that breeds can be incorporated. Further, here I have shown that Avishan, it is a prolific three breed cross where farmer can get 1.5 times income without extra investment. So in investing the same amount of money, So it is a prolific breed of sheep. The twinning percentage is more in this breed. So we know that sheep generally gives the birth to the only one lamb at a time. But this breed, if we can change this the breed, so the productivity of the system will enhance 1.5 times. So these kind of breeds and technology also available with respect to Indian condition. Also, I already am showing the here, and like Nari Suvarna and Nari composite breed. So these breeds are from Maharashtra. So they have incorporated the FAC-B gene from Garol breed, and these breeds are giving the more twinning to the farmer. So what I mean to convey here, that these are just the examples. In case of poultry, in case of the goat, in case of the sheep, in case of the dairy, in case of the buffalo, more and more efficient breeds should be discovered and given to the field and given to the system, so that we can have a very good sustainable system which can sustain and produce at the maximum and optimum level, and which also reduce the negative impact on the environment. Next, please. Further, the production of Dumba sheep, that fat tail sheep, seem to be one of the options for enhancing meat production and bridging the gap between the demand and availability of meat in the country. Next, please. So here in brief, we have talked about the strategies which are available. There may be several more strategies, but we have, due to the limit of the time, we have discussed very important strategies here. So, but for adoption of these strategies, there are some challenges and some barriers also. So, for example, to change any system, we have to have incorporate some initial cost. So the initial cost, for example, I have told about the precision livestock farming system. But to purchase that system, to install that system in the farm, it will incur some cost. So this initial cost, who want to bear that cost? Because the farmer, the producer will get the same price. So why he will incorporate that cost? That is the challenge and that is the barrier. So, and the knowledge and training. So there is lack of knowledge about the sustainable practices. So we have to disseminate the technology available to the producers, to the next level, to the field level, so that more and more sustainable technology, which are very common but not in the use, can be incorporated, so that to improve the efficiency of our system. Market access. Presently, in developing countries like India, there is less kind of consumer demand for these sustainably produced livestock products. So market access is very limited. If we can say that in metro cities or big cities only, there is such kind of demand. So where consumer are demanding the product which are produced from a very good farm, like organic farm. We can say organic farm is opting the sustainable practices. So this demand of these kind of product is very less. So policy support also. So it needs policy support. You have to give some kind of subsidy. You have to create some kind of market. So these are the barrier at present for adopting these sustainable technology. But in future, definitely we have to overcome these barrier. We have to think about these barrier, because the if we go like this, we are not going to sustain in long term. So we have to change our system. We have to incorporate these strategies and convert our system to the sustainable one. So here I come next, please. Here I can come to conclude this talk, that, and we in conclusion we can say that livestock sector presently facing several challenges and pressure as well. So considering the importance in economy and livelihood support, sustainable strategies need to be adopted. But while adopting the strategies, challenges which I discussed, that need to be addressed properly. But in summary, We can say that sustainable management of livestock and poultry is essential for balancing the growing demand for food with the need to protect our planet's resource. Thank you so much.
Thank you, sir. Thank you for your amazing presentations, and you have thrown light on a very important topic, which I must say, and I'm just waiting from the participants in the chat box. And for until I wait, I will ask you some questions which I have got. So, sir, as you said that livestock sector is facing challenges and pressure. So, how the government, how helping
Yes, ma'am. Very rightly asked question. Actually, as I said, that in or the USA or say the Australia, New Zealand, these countries now they have made the commission for that. So they are creating some kind of regulations. They are creating some kind of rules, and they are also providing the policy support and also the market, because market is very necessary. Why producer will change to a practice which is costlier than the existing practice? Because it is benefiting the environment? No, he will see his benefit only. So at present, I must say that in our country, these kind of policies, these kind of support need to be given to the producer, so that they will be motivated to adopt these sustainable strategies, like to reduce the methane emission. Nobody, no producer is considering about the methane emission, but government should come forward and say that we will give you, we will provide you this kind of support if you produce your product with less emission. So this can be done.
Yeah, there should be some kind of incentive for
Yes,
farmers,
yes,
for the
yes, exactly,
producers, so
exactly.
that they can
But
feel
at present, you
motivated.
ask, no, there are no kind of incentive for these kind of strategies, because we are still developing and we are still behind the production.
Yeah, actually, like we are facing a problem of stubble burning in some of the northern states of
Exactly,
India.
exactly.
So there are some incentives for farmer who don't burn the stubble, and so
Yes,
it
yes.
helps the environment and it motivates the farmer to do such kind of things. So how field veterinarians can play a role in doing all these things, like, or how they are doing? Actually, I don't know whatever they are doing. So what are they doing at ground level?
Yeah. Actually, field veterinarians have very much role in this kind of practice. Because the one important issue with the livestock production in India is the antibiotic residue also. So for the field veterinarian, they are directly concerned with the treatment aspect. So while they are giving some kind of antibiotic or suggesting some kind of drug, they should very restrictly communicate these drugs to the farmers, because if you use it judiciously only, then only the residue part will be lesser in the our product. If we see our milk, we are, although we are highest milk producer country, but unfortunately very few amount of milk out of this 240 million ton milk will meet the standards of the developed countries, European countries. Why? Because they are having high amount of residue. So that can be used in our country only. We cannot export that product. So that is the problem. So if we can export, it will give the incentive. So because of these practices, we are not able to do that. So field staff is having very role in because they must be aware. They should be motivated. They should be encouraged to do these kind of things.
Yes, sir. And one thing, the nanotechnology, it is getting more and more popularity nowadays when we talk about the research perspective and when we see the
Yes, yes.
literature. So on ground level, how nano formulations are playing a role in this livestock health management? Is there any like in practically these are being used or not now?
Nanotechnology are being used in the kind of drug delivery system. Like if we see the mineral mixtures, nano formulations are available, nano zinc are available. So, but very limited they are being used. Still, it is in the research phase actually, not coming to the ground level. Still, we are researching on this whether we have to use it or not. But definitely, nano formulations have their own benefits because of their availability. They are more and more available. So that's why definitely they will help in improving the efficiency of production of the animals. So that is clear. But when that will come into the field, that will time will decide.
Yes, there one last question I would like to ask. Like you have worked on the conservation of various indigenous breeds of animals. So how do you see the future of this conservation? Like it's very important. We need to conserve our indigenous breeds of animals. So how do you see the future, and what are the challenges that you have faced in this process, so that our
Yes,
part
ma'am.
parents can know and they can get inspired from your
Yes, yes.
work.
Yes, yes. You are very rightly said, ma'am. I was involved in the conserving and registering 3 breeds of the goat in our country. So I was very fortunate to work in this area and I have seen the changes. For example, recently in January 2025, we have registered the Bundelkhandi goat of this Bundelkhand region, falling in Jhansi and Datia, Jalon, and several districts are there. So I have seen the change, like the NLM scheme before it was working for which is providing the loan to the goat farmer who are goat entrepreneur actually for their business. So previously they were not able to use this goat, this indigenous goat, because this was called as the local goat. But now after registering this breed, after defining this breed, after conserving this breed, they will be able to get the loan for their own goat, for their local goat. Bundelkhandi goat. So they are very much appreciating this. So many farmers came to me and they appreciated that, sir, we are now able to rear our own goat. We don't have to go to Rajasthan to purchase Siroi goat or to the Etawah to bring some Barbary. So we can have our own. Previously, they were not able because government policy was like that, ki only registered breed you can use in your project. So this one change, one example you can see that, and we if we give the value to our breeds, no, we have potential breeds, there is no doubt about that, but we have not yet recognized. I think in my experience we have seen 3 breeds, and after their recognition, they have slowly because it is not like all of sudden all things will become changed, but slowly the benefits will be visible, and that definitely will come.
Thank you, sir. It's indeed a very
One more question from the participant.
Yes, yes.
Dr. Deepak,
Yes.
as we are slowly shifting towards a stall feeding system
Hmm.
of goat, but it costs more than traditional one. So what do you think that there should be a minimum support price MSP in livestock also?
A very good question. I think this is because I told you that initial cost, whenever we shift, that will cost the initial cost. And only if premium prices are paid for that products, then only that system will survive. Suppose that if we are doing a goat farming and if you are doing a commercial goat farming. So in traditional system, what you will do in morning, you will send all the goats to a grazing and in evening you will tie them. But in commercial system, you will have to bear the cost of the feed also, and definitely the quality of your product will be more if you are following all the scientific practices, you are doing the deworming, vaccination, timely treatment, you are allowing for the animal welfare also. So that quality of the product will be definitely more than the traditionally produced product. But here the differentiation is not there, the branding is not there. So definitely the minimum support price, in my opinion, definitely not kind of minimal support price, but premium prices for these kind of products which are coming. So this is called traceability. So traceability is not there in our Indian system. From where this product is coming, you will not knowing. If you will purchase a packet of milk, only brand will be written. From where it is coming, from which farm it was produced, you will not be knowing. So that system, when it will come, It is already existing in the developed country. So the premium price will be given. So for this, we will have to use the technology. But for technology, cost is involved. So all cycle is there. So if we complete this cycle, initially we have to invest. And investment first will come from the government side, definitely. So in my opinion, MSP, I don't think, but premium prices should be given.
Okay, okay, thank you.
Thank you, sir. Thank you so much. You have delivered such an excellent and great presentation, and I must say that you are doing such a great work, and you are working for the farmers, and for the animal growers. You are working for them, and we wish you all the best on the behalf of the
Thank you.
organizers, the participants, and on my personal behalf. We wish you all the best to continue working like this in future as well, and for helping the farmers, and for more and more research work in this field. Thank you, sir. Thank you so much for your
Thank you.
precious time.
Thank you,
Thank you
Dr.
so much.
Deepak.
Thank you. Thank you to all the organizers. Thank you, sir. Thank you so much.
Thank you, sir. Thank you so much. So, dear
Thank you, Dr.
participants,
Deepak.
it was a wonderful presentation and wonderful talk with Dr. Deepak. And now we are moving forward. So, maybe moving forward, I must say that we are highly privileged to have our another guest, Dr. Ajay Kumar, with us. So, before inviting Dr. Ajay Kumar, I would like to let you know about sir a little bit. So, Dr. Ajay Kumar is a senior scientist at the Division of Biochemistry, IBRI Ijatnagar, Bareilly, and the primary research work of Dr. Ajay focus on developing diagnostic on BHV1 virus infection in bovines and understanding the biology of latency reactivation cycle of this virus. Additionally, sir has worked on developing a colonization comprised mutant of Salmonella typhimurium and has executive hypothesis involving codon deoptimization of the anorbic regulator of Salmonella to decrease its virulence. Sir has published over 40 research papers in peer-reviewed journals, and Sir has completed a one-year postdoctoral fellowship at Auburn University, Alabama, USA. Apart from research, Sir has organized various international training courses under the TCS Colombo plan and a DST SERB sponsored workshop. Sir has successfully completed 5 DBT and DST funded projects as the principal investigator, and currently leading one DST and one DBT project under his leadership. So, sir, I must say that your achievements are endless, and we cannot enlist all of them in this short span of time. So, I humbly request you to take over the stage and tell the participants about today's topic. So, the topic which sir is talking about today is the role of molecular markers in in the investigation of livestock diseases. So, I request you, sir, please take over the stage.
Okay. Thank you very much. Can you hear me?
Yes, sir.
Can you hear me now?
Yeah, sir, but your volume is a little bit low.
Okay. Now can you hear?
Sir, I can hear you, but the sound is a little bit low, like you are not properly audible. I think you need to speak a little bit louder, or there is some.
Now can you listen me?
Yes, sir, you're audible.
Okay. So thank you very much. Actually, I have some throat infection, so please bear with me.
Okay.
So right now, can I share the slides?
Yes, sir.
Okay.
Yes, sir, we can see the slides.
Slide is moving, no?
Yes, the slides are moving.
Okay. So very good evening. I'm Dr. Ajay Kumar. I'm here going to discuss about molecular markers. What are these molecular markers? How they have effect on different livestock diseases, and what are the techniques that we are going to use to detect these molecular markers. So we know that these molecular markers are specific biomolecules. It could be your carbohydrate, lipid, protein, nucleic acid. So these have they can cause genetic mutations, gene expression changes, or protein alteration associated with the diseases. So what are the molecular markers in disease investigation? One is your DNA markers. These identify genetic variations linked to diseases. Second one is the RNA markers. These reflect gene expression. We know that DNA is converted to mRNA, and mRNA is responsible for production of protein. So all mRNA is not converted to protein. So there are some mRNA which has some expression which we call it post-transcriptional modifications. So all mRNA not converted. So they have some implications on the diseases. The third one is protein markers. We detect abnormal proteins, suppose tumor marker proteins. Sometimes we also see different proteins like serum enzymes like ALP, AST, SGOT, SGPT. So different LDH, they are protein markers which helps in the investigation of diseases in the livestock. Now coming to the applications of these molecular markers. So they have application in the disease diagnosis, prognosis and risk assessment, treatment guidance, what kind of treatment we can follow. Disease monitoring means progression of the disease, and infectious disease detections like molecular markers detect viral, bacterial and other pathogens. Rapid diagnostic tests that we use for these markers for early detections. Now coming to one by one, what are the DNA markers? So DNA-based molecular markers help identify genetic variations. Okay, so common DNA markers are, we know that single nucleotide polymorphism means they change in the single nucleotide that can be detected by different techniques. Coming to microsatellite, this microsatellite helps in knowing that we use it RFLP DNA fingerprinting, that differences in the microsatellite numbers, we find the differences in the genetic makeup. Now coming to copy number variations. So there are some duplications or deletions of the DNA segment that will result in the neurodevelopmental disorders and cancers. Then mutations, deletion or additions of the nucleotide helps in the mutations. Just like BRCA1 means breast cancer genes, there will be mutation helps in the development of breast cancer. Now coming to RNA markers. So RNA markers provide insight into gene expression and disease progression. They are crucial in detecting infections, cancers, and neurological diseases. Some common RNA markers that we use it, mRNA markers, that messenger RNA, microRNA markers, because there are some microRNA present inside the cell which regulate the expression of mRNA. Then long non-coding RNA. These are the non-coding sequences which is present inside our DNA. So these RNA are responsible for regulation of the gene. These long non-coding RNA they could be the part of the silencer repressor genes. Now coming to the protein markers, protein-based markers help detect abnormal protein production, post-translational modification, and enzyme activity related to the diseases. So some common protein markers, that tumor markers, elevated levels of protein like CA125 ovarian cancer, prostate-specific antigen, their prostate cancer, then alpha-fetoprotein, which is used in liver cancer. Some enzymes are there like ALT, AST, SGO, then your alkaline phosphatase, GGT, LDH. These are the enzymes which are having clinical importance. They are present in the serum. They have no action on the serum. They are coming from the damage to the organs. Suppose ALT, AST, they are coming from the liver damage. If there is damage, fibrosis or cirrhosis, hepatitis of the liver, so these enzymes are coming into the serum. They help in the investigation of the disease of the liver. Then cytokines and other growth factors, they are present in the serum. They help in the identification of inflammatory disease like CRP, C-reactive proteins, which is highly increased in inflammatory diseases. Another marker is the epigenetic marker. So these epigenetic changes regulate the gene expression without altering the DNA sequences. These modifications play a crucial role in cancer, autoimmune diseases, and neurodegenerative disorders. So some epigenetic markers are DNA methylation. We know that methylation play a very important role in activity of the gene. If there will be methylation in the promoter region, so what happens? Promoter is methylated, so promoter gets least, means they are getting down. So expression of that gene will be goes down. So histone modification, histone acetylation also linked to leukemia and other cancers. These are epigenetic modifications. Now coming to, as we have seen, that molecular markers can be analyzed through two ways. One is at nucleotide level, that will be through identification of bacterial or pathogenic nucleic acid by PCR, real-time PCR, quantitative PCR. Then another level is at protein level detection. Use of protein either as a diagnostic purpose. Suppose we are having a lot of diagnostic tests, ELISA-based test or lateral flow-based test. They are using the protein as a diagnostic purpose. Then for prophylactic purpose, same sometimes protein that are immunogenic in nature, like subunit vaccine, killed vaccine, mRNA vaccine, protein vaccines that we are using that can be used as a prophylactic for disease investigation. Then if you are coming to one by one, what are the different tools that we are using for identification of these molecular markers? So first we are coming to identification at nucleotide level. So when we are identifying them at nucleotide level, so we first go to the most common technique which has come into the existence in early 1990s. So PCR, polymerase chain reaction for identification of pathogen. So this PCR is an in vitro technique. Normally what happens in our cells, DNA is getting doubled. So that DNA is getting doubled by DNA polymerase. We have different DNTPs are present in our system. So same what we are replicating in outside in the tube system that we call it polymerase chain reaction, which is in vitro technique for amplification of a particular region of DNA. So what we do here, suppose you are going to identify Salmonella bacteria. So, or you are going to identify a virus, bovine herpes virus, you are going to identify a Newcastle disease. So what we take, a particular gene is taken. From that gene, we are identifying particular gene sequences. We are designing a primer. A primer sequence will be there. This primer, which is a complementary sequence to that sequence. And this primer, generally, they are 18 to 25 nucleotide length, single-stranded, and complementary to both the sequences. One is coding sequences, one is non-coding. We know that DNA is double-stranded. So one sequence is coding strand, another is non-coding strand. So this primer, a pair of primer that we are using, they are complementary to both the coding and non-coding sequences. So these oligonucleotide primers are used, they bind to the template, and then we use a DNA polymerase. Here, DNA polymerase that we use is Thermus aquaticus DNA polymerase. They are heat resistant, so they are generally working at 72 degrees centigrade, their optimal temperature. Because every enzyme has their optimal temperature, optimum pH where they work maximally. So these new strands have defined 5' ends where the 3' ends are potentially ambiguous in length. So what happens in polymerase chain reaction? What happens? The primer which is binding. So every cycle, the template sequences, they are getting double of number. So if we see that every 10, suppose you are taking one template. So at 2 to the power n, the number of cycle, n is the number of cycle. If we are going to the 30 cycle, we are getting 1 billion template produced through this polymerase chain reactions. So what happens? There are 3 steps of polymerase chain reaction. One is your denaturation, second is your annealing, third is your extension. So denaturation is the temperature where both the DNA gets denatured, means their hydrogen bond formed between these two strands are getting broken down. So this generally happens at hitting the DNA at 94 degrees centigrade. You can see here. Then annealing. So binding of the primer to the template strand. So this happens generally, we use 55 to 60 degrees centigrade. Then comes your extension. After binding the primer to the template, what happens? So DNTP is added as per this stacked DNA polymerase has somewhat 5' to 3' exonuclease and 5' to 3' your polymerase activity. As per that polymerase activity, they add DNTP to the primer sequences and there will be synthesis of new strand. So after 30 cycles, we are getting billion product are formed. So generally in polymerase chain reaction, we run for 30 to 35 cycles. So we are getting billion product formed. This billion products is in a sufficient number that can be detected in agarose jet electrophoresis. So PCR reaction that we see that contains a target DNA template strand to one set of primers, one is forward primer, one is reverse primer. Then we take thermostable DNA polymerase, we take nucleotides DNTP, then another we take magnesium ion. This magnesium ion is used as a cofactor to this DNA polymerase. So what happens? DNA polymerase works, they need a cofactor as magnesium ion. If magnesium ion is not present, so what happens? DNA polymerase will not work. So DNA polymerase is a holoenzyme, apoenzyme and as a cofactor magnesium ion. Then they undergo denaturation, annealing, and extension. So what are the different applications that we use for PCR in different diseases? So the genetic disorders, if we can detect mutation in hereditary disease, the cancer detections, some cancer specific markers that we can take like breast cancer gene, the neurological diseases, if there is some changes in the enzymes of some disease like Huntington's disease, the infectious disease detection like viral diseases, the hepatitis, then SARS-CoV, then human papilloma virus, then bacterial infections like tuberculosis, Lyme disease, MRSA, the parasitic diseases, fungal diseases, the forensic science and DNA fingerprinting that we use in PCR, like criminal investigation that amplify DNA from crime scene, paternity testing. Other applications include genetically modified organism detection. Suppose we are going to generate a genetically modified organism like salmonella. This salmonella can be detected by polymerase chain reaction. Then phylogenetic studies like analyzing DNA sequences to study evolutionary relationship. The cloning and gene expression studies we can do by PCR. Then epigenetic study like DNA methylation can also be studied by application of PCR. So we know that PCR has 3 steps. One is your denaturation, then annealing and extension. What happens? We know that there generally we use 2 kinds of PCR. One is traditional PCR that we use. Then one is real-time PCR. So nowadays real-time PCR is more sophisticated, helps in diagnosis of diseases. So PCR has 3 forms. One is exponential phase, then second is linear phase, then plateau phase. In traditional PCR, what happens? The detection of the particular disease or by amplification, then it's plateau phase. When the PCR is run at 30 or 35 cycles, we are running in the agarose jet electrophoresis. So we do not know at what cycle of PCR we are getting double the number of the template DNA. So to avoid this technique, so what we do at real-time PCR. This real-time PCR, what happens? They are working on exponential phase of the cell. So this exponential phase where every cycle our template is getting double the numbers. So how it is getting doubled? This real-time PCR, that is the more advanced technique of the normal PCR. The principle of real-time PCR works on use of fluorescent dye. So the amount of nucleic acid present into the sample is quantified using the fluorescent dye or using the fluorescent labeled oligos. So there are 2 chemistry. One is DNA binding dye, one is DNA hydrolyzing dye. So DNA binding dye that we normally use cyber green chemistry. So this cyber green, which is binding to double-stranded DNA. So more the amplification, more the cybergreen will bind to the double-stranded DNA, more the fluorescence will be detected. So this cybergreen chemistry works on more the amplification, more the fluorescence, more will be the number of template. So DNA binding dye method is best technique for real-time detection. So this dye has its own fluorescence. Once the dye binds to the double-stranded DNA, the fluorescence emitted by the dye increases 100 to 1000-fold than the original signal. When the dye is not binding to the double-stranded DNA, they have very less fluorescent signal, which is undetectable. So however, the original dye fluorescence is taken as the baseline for the detection. This baseline, we call it the CT value, the threshold cycle. This method is rapid, quick, reliable, and cost-effective, where we do not need to run the agrogel electrophoresis to see the amplification of DNA. We simply see in the real time when the fluorescent dye is coming or exceeding the baseline or threshold cycle, threshold line. Another dye which works is your probe based, that we call it Tachman probe. This is Tachman probe where it have two kinds of fluorescent molecule bind. One is your fluorescent dye, one is your we call reporter dye, one is your quencher dye. This green that you are seeing here is a reporter, a black is your quencher. So this probe is a 25 to 30 nucleotide length primer where two fluorescent molecules is attached. One is reporter, one is quencher. So when they are close proximity, there will be no fluorescence because quencher will take the fluorescence and there will be no fluorescence. So when it is binding, when there will be amplification, this probe will be hydrolyzed by 5' to 3' exonuclease activity of the DNA polymerase, and this probe will be broken down, and then quencher and reporter dye will be released. Once it is released, there will be no quenching by the quencher. They come into the solution, and then there will be fluorescence. So more the amplification of the DNA will be there, more the hydrolysis of the probe, and more will be the fluorescence. So this probe-based chemistry works on the hydrolysis. So this Tachman probe-based chemistry of the real-time PCR is very specific and better, having high sensitivity than your cybergreen chemistry. So what are the application of this real-time PCR? So quantification of gene expression. We can quantify, like suppose we are going to identify the C-reactive protein which is used in the inflammation, or TNF-alpha, different cytokines, different interleukins. So one is your disease condition, one is in your normal diseases, or one is your normal life cases. So expression level of these cytokines can be studied by real-time PCR. Quantification of viral or bacterial load. How much bacteria or virus is there? We have seen in the corona time, the real-time PCR was doing good in identifying this COVID-19 load in the nasal swab or in the body. The validation of microarray. We know that microarray is the chip where a lot of genes are impregnated on that chip. So how the microarray can be validated? A particular gene is expressed, that gene can be used in real-time PCR. Detection of pathogen, whether pathogen is present or absent. Genotyping. So genotype is about single nucleotide polymorphism is there. By real-time PCR, we can identify the response to treatment. So suppose we have given antiviral treatment, antibacterial treatment, how the bacterial or viral load has gone down. Now coming to detection of molecular marker at protein level, the different systems are there, like Western blotting, ELISA is there, immunohistochemistry or immunocytochemistry, flow cytometry, and later-based flow-based point-of-care techniques. Now coming to Western blotting technique, we know that there are different blotting techniques are there, like Southern blotting for DNA identification, then Northern blotting, RNA identification, then Western blotting for protein identification. So Western blotting is an immunoblotting technique. Immunoblotting is we need to have antigen, antibody, primary antibody, and secondary antibody. This Western blotting is immunoblotting technique. which rely on specificity of bonding between a molecule of interest and a probe to allow detection of the molecule of interest in a mixture of many other similar molecules. In Western blotting, the molecule of interest is a protein, and the probe is typically an antibody raised against that particular protein. Before that, we need to run the protein through SDS-based electrophoresis to separate this protein. So what are the steps involved in Western blotting is first sample preparation. So you need to have a sample, like it may be tissue sample, cell sample, or bacteria or virus infected tissue sample. Then take the protein out from the sample and run in the gel electrophoresis. And then after electrophoresis, transfer this protein which is present on the gel to the membrane. That membrane could be a nitrocellulose membrane, or that membrane could be a PVDF membrane. Then we do blocking, because where protein has not bound, that area should be blocked by use of bovine serum albumin or skim milk powder lactic albumin, because they are neutral protein. They will bind to block the non-protein bound places. Then we are using antibody. So what happens? We use two types of antibody. When we are going to identify, suppose we are going to identify a particular bacterial protein. X protein is there in the bacteria. We are going to identify this bacterial protein. So we have anti-X antibody. So this antibody is raised against bacterial X protein. So this antibody will bind to the X protein present on the blot. We have transferred from the gel to blot. Then we use secondary antibody. Secondary antibody will be species-specific antibody. Suppose primary antibody, which is raised against X protein, is raised in, suppose, rabbit. Secondary antibody will be anti-rabbit IgG. That will be labeled either with the HRPO, horseradish peroxidase enzyme, or alkaline phosphatase. Then we add the detector system. Like if we are using a horseradish peroxidase, so we use DAB substrate, H2O2 and DAB, that is diaminobenzidine. They give a colored precipitate. So at that position, by use of protein molecular weight marker, we know that suppose X protein is 50 kilodalton. At 50 kilodalton, we are getting, we can see here in the Western blot, we are getting a band. This band is of suppose X protein band. So presence of band of this protein tells you that yes, bacterial protein is present in the tissue sample. So this helps in your diagnosis of bacterial through a protein labeling system. Second one is coming your ELISA, that we use enzyme-linked immunosorbent assay. Very common technique nowadays people are using for diagnostic purpose or different application. So this is just a variant of radio immunoassay. For that, Yellow and Birsa got Nobel Prize in 1960. They have developed the radio immunoassay. In 1971, Perlman and Ingwell, they have developed this enzyme-linked immunosorbent assay, which is a derivative of radio immunoassay. What happens here? Either antigen or antibody is coated on the 96-well ELISA plate. So this antigen or antibody which binds with the ELISA plate by hydrophobic interactions on the plastic surface. This antigen is, if we are coated the antigen, so this antigen is known. We are going to identify antibody which is present in the serum sample. So serum sample is taken, which is added to the well where already antigen is there. This antibody, if against that particular bacteria, so this will bind to the antigen. So antigen antibody complex formation will be there. Then we take secondary antibody, just like your Western blotting we have done in the blot or membrane. Here we are doing in the liquid system. So we take a secondary antibody, which is again enzyme linked or conjugated. Then there will be additional substrate and formation color product will be there. So we can see that different color form, because in Western blotting, this is a qualitative test. It will give you yes or no. They will not give you how much bacterial or viral load is there, presence of protein. But in ELISA, we can quantify how much load of bacteria or virus through that protein we can identify. So There are different variants of ELISA. Is there direct ELISA, which helps in the detection of bacterial or viral antigen. Indirect ELISA for antibody present in the serum sample. Then sandwich ELISA, either it could be antigen or antibody. Then competitive ELISA for detection of, again, antigen or antibody. So what are the different applications of ELISA? Because ELISA is a very sensitive technique, because we know that Western blotting cannot be used for the quantification. So ELISA can be used for quantification or presence of, or we can use the serosurveillance, serum antibody concentration, detecting potential food allergens. Presence of allergens in the food can be detected by ELISA also. Disease outbreaks, because in that particular area, if particular disease is there or not, that can be detected. If, because Western blotting can be done, it is a cumbersome technique. But in ELISA, we can do directly by presence of that particular pathogens. Detection of antigen can be done. Detection of antibody in the blood samples for past exposure to diseases. ELISA also helps in differentiation of early infection and late infection. We know that in our body, there are two kinds of antibodies are present. One is IgM, one is IgG. When there will be early infection, then as IgM will be detected, their level will be very high. This starts from 3 days or 4 days of infection and it persists for 4 to 8 weeks. Then after that, IgG take over. So IgG will tell you the presence of disease in that particular region, and IgM will tell you immediate presence of that disease. So based on IgM antibody and based on IgG antibody, we can identify whether infection was early in that particular region or in that animals, or infection has taken place long back. So that presence of IgG will tell you that infection was there. Now infection got rid of, and then only IgG. So it will tell you the disease surveillance, or we can say zero surveillance of that disease. Then coming to immunohistochemistry. So immunohistochemistry or immunocytochemistry, this is also a variant of a kind of immunotechniques where we are using a primary antibody, secondary antibody that is based on the tissue sample. There no need to take the protein out from the tissue, or no need to take the serum samples. Take the tissue and you can identify the presence of that bacteria or that virus in that tissue. How much load? You can see here different brown colors. The brown colors tells you that presence of bacterial pathogen or viral pathogens and presence of primary and secondary antibody. We use primary and secondary antibody. These brown precipitates tells you that yes, these bacteria virus are present, or load of this bacteria virus in that tissue sample. Suppose this can be a liver, or this could be your kidney, or this could be of some other tissue samples. Now coming to another technique, flow cytometer. The flow cytometers, they are simply the measurement of cells when they are in flow. So what we do, we know that every cell has their receptor. We know the cells, that one cell is CD3 cells, T cells, B cells, or T helper cells, T cytotoxic cells. How we recognize that these are T cells or these are B cells? So these cells are having their particular receptors. So for example, you can see here T cells. T cells has CD3 receptors. CD3 is a kind of glycoprotein present on the surface of the cells. So B cells, they have CD19 receptor. So B cells is responsible for production of antibody. NK cells, natural killer cells, they are having CD56 receptor on the surface. So these receptors are different receptors. Or we can say that HER2 receptor present on the breast cancer cells. So HER is human epidermal growth factor receptor 2, which is only present on the breast cancer cells when the breast cancer cells are undergoing metastasis. They have high level of HER2 expression. So in flow cytometer, what happens? Again, we take antibody. That antibody is conjugated. Earlier, we have seen that this antibody is conjugated with enzyme. Here, antibody is conjugated with a fluorescent compound. See this fluorescent compound, that could be FITC, fluorescent isothiocyanate, or it could be APC, L-phycocyanin. So this fluorescent compound will be tagged with the antibody. When we are taking the cells and we are adding the antibody, this antibody binding to the cells, and these cells can be run in the flow cytometer, and flow cytometer will detect you how much fluorescence is getting emitted from the cells. So they have fluorescence detectors. So this FL1 that we are seeing in the slides, FL1, this FL1 is a fluorescent detector which detects the FITC. For L-phycocyanin, we have FL4 detectors. So they detect only L-phycocyanin. So based on this flow cytometer helps in detection of these particular cells which are having high expression of HER2 cells. So we have seen here there are different kind of breast cancer cell line, MDA, MB, MCF, MDA, MB453, SKBR. These all are human breast cancer cell line. They have high expression of HER2 expression cells. So more expression that are detected by anti-HER2 antibody labeled with FITC can be detected by flow cytometer. So these are also a technique. So it helps in detection of your cancer cell line, metastatic cancer cells with the help of HER2 receptors. Here we can also see in some disease condition, we know that there are T helper or T cytotoxic cells. T helper cells are responsible for humoral immune response, and T cytotoxic cells are responsible for cell-mediated immune response. So this humoral immune response, this T helper cells have CD4 receptors, as T cytotoxic cells have CD8 receptors. So we are really anti-CD4 and anti-CD8 antibody labeled with two different fluorescent compound, FITC and L-phycocyanin. So these two different antibody added to the blood cells, and we see the different, this is the normal persons, where we see that this, you can see the quadrant 4, this is the lower right corner. You can see the lower right corner, they are only CD4 cells, and the upper left corner, they are only CD8 cells, and the lower left corner that you see in the Q3, they are neither CD4 nor CD8, they are other blood cells, and Q2, the upper right corner, they are the double positive cells. So you can see here in this population, CD4 is more dense populated. But when you are seeing the infected population, the CD4 population is getting down. So this helps you the identification of molecular marker in the form of CD4 and CD8 in the blood with the help of flow cytometer. This flow cytometer can also be used for detection of your natural killer cells, which is also a prognostic marker for immune cells, then neutrophils, basophils, eosinophils, then memory cells, then NAV T cells, different immune cells, or even sometimes mesenchymal stem cells can also be detected by flow cytometer by using their own circuit, because mesenchymal stem cells also have their CD34 markers. So we use antibody against CD34 run in the flow cytometer, and fluorescence will be detected, and this fluorescence will tell you how much mesenchymal stem cells are present in the bone marrow. Another, is it visible to you? The slide is visible?
Yes, sir, slides are visible.
Okay, so another technique that we use to identify the protein, we have seen that protein, either nucleotide or protein markers. So nucleotide we have seen by PCR, real-time PCR, a protein by Western blotting, immunohistochemistry, ELISA, flow cytometry, and another one is lateral flow assay. We know that lateral flow assay is a point of care diagnostic technique that we use generally. The most common that, just for example, we are giving that in pregnancy diagnosis, we use that SCG, human chorionic gonadotropin, that is present in the urine sample. So in lateral flow, there are different systems are there. One is sample pad, conjugate pad, test line, control line, and absorption pad. So what happened In sample page, we are adding the sample. Suppose we are going to identify a particular bacteria like Salmonella. So we are going to identify whether Salmonella is present or not. So we take the sample, we put the sample on the sample pad. So what happened in this sample will move to the conjugate pad. You can see here conjugate pad. And this conjugate pad contains antibody against your Salmonella antigen. And this antibody will be labeled with either gold nanoparticle or biotin level particles. So once the sample contains the Salmonella antigen, this antigen will bind to the antibody against that Salmonella antigen. If binding will be there, and there's gold nanoparticles will be there. Then another test line. So we have here test line and control line. Control line contains the antibody against this antibody which is present in the conjugate pad. So control line contains the antibody against the conjugate pad, and test line contains the antibody again against the Salmonella antigen. So two kinds of antibody. One is present in the conjugate pad, which is labeled with the gold nanoparticles, and test line contains the antibody which is not conjugated. If sample contains your Salmonella antigen or any bacterial viral pathogen, so this will bind to the antibody. After binding through this antibody, they move to the test line. Test line also contains the antibody against your Salmonella. So two kinds of antibody you can see here in the picture. This is an antigen, two antibody. After binding of two antibody to this antigen, so gold nanoparticle will have some emission spectra. This emission spectra will give you a colored product. So test line will have colored product. In the control line is also bound to the against the antibody present in the conjugate pad. So this is antibody, a control line anti-antibody. So there will be also interaction of the antigen, antibody and anti-antibody. So there will be formation again color product because this antibody is labeled with the gold nanoparticles. But if antigen is not present, so what will happen? Test line will give you colored product because test line contains the antibody against this antibody. So color will be there. But since the sample is not containing your antigen, so there will not be antigen antibody complex formation in the conjugate pad. So there will not be formation of colored product. So control line is giving you a idea about, yes, your test is working well, and present or absence of antigen will be told by this test line. So this test line is very important. So here we can see here in this diagram how HCG antigen binding to the antibody, which is labeled with the gold nanoparticles Orium. There is flowing to the test line. Test line again contains the antibody against the HCG and protein. Then binding interaction, there will be color formation. Then because antibody they are more in number, they are moving towards the control line. This control line without presence of antigen still they will give you color. So we have seen that two kinds of means we have molecular markers in the form of nucleotide, in the form of protein. Different techniques we have employed to identify these molecular markers in disease condition. Either it could be a bacterial viral pathogens, it could be a genetic diseases, or it could be a cancer cases. So this is all about different conditions, different techniques, and different molecular markers used for livestock diseases. Thank you very much.
Thank you, sir. Thank you for your presentation, and we must appreciate and we must thank you for your dedication and commitment as well. That despite having throat infection, you delivered the lecture very enthusiastically. Thank you, sir. Thank you so much.
Thank you, sir.
Okay, thank you very much.
Thank you, sir.
Thank you, sir. You accept my invitation, although you are suffering from throat infection, but you deliver nicely 1 hour to our audience. Thank you, sir, very much.
We are very thankful, sir, for your dedication. And we are thinking that we should not take any questions because you are suffering from the throat
Exactly.
infection. And if we get any question from the audience, we will send the question to you through mail or some electronic mode, and then we'll get the answers. Is it fine, sir?
Okay, fine, fine. No issue.
Yes, sir. Thank you for coming, sir, and we wish you great health and take care of your health.
Thank you. Thank you very much.
Thank you, sir.
Thank you so much, sir. So, dear participants, as we have reached to the end of today's session, so on the behalf of Vital Biotech Agriculture Forum for Technical Education of Farming Society, Kota, Faculty of Veterinary Science and Animal Husbandry, Ranchi, Birsa Agriculture University, Ranchi, and the entire organizing team, I deliver my heartfelt gratitude to our amazing speakers of the day, Dr. Deepak and Dr. Ajay, for their insightful talks and for their unwavering support to the organizers, for their commitment and for their hard work. Their hard work and experience, I'm indeed sure that must have motivated and boosted the knowledge of our participants, and you must get inspired from their work. So thank you to the speakers, and thank you to the participants for joining this 7 days training program, and for being so patient and attentive throughout the session. So thank you all and have a great evening.
Thank you all.